As Janet said, my name is Amanda. I'm the outreach coordinator for the foundation, but most importantly, I'm the mom to Ripley. She is 7 and she has Kabuki syndrome. I want to thank you for being here today and being willing to learn. You can learn the science and you can do something. We're building an educational pipeline to go alongside the therapeutic pipeline to make sure this research is accessible. So you don't have to wait and watch. You can find a way to drive research with us. And as families, if we can learn the science of Kabuki syndrome and understand the research pipeline, we are better able to advocate for, accelerate, and participate in the research that can change our lives. If you know your child or yourself or your disease, you know you can ask better questions and get better care. But you can also use your understanding of your disease as the foundation to learn about the research that leads to treatments. We want to deliver that education to help you understand not only the syndrome, but how its symptoms could be treated with more research. So during the breaks today, you can download the educational content we've created so far and really take your time with it when you have time. You can share it with your friends, families, doctors, and I want you to send me your questions and suggestions. These are the first phase of the education that we're providing to empower you to drive research with us. And as we look at the role of the researchers, the foundation wants to make sure we're all asking the right questions before we go looking for the answers. Later in the scope of science, we're looking at what published science exists and what is missing. With crowdsourcing surveys, we're identifying what is missing from the family's perspective too. What are you missing? But what are you interested in doing to change that? You can influence clinical trial design, and we can educate and amplify your voice so it gets to the researchers when it counts. We are lucky to have a handful of researchers dedicated to Kabuki syndrome, but we have to understand that their research cannot move forward without our participation. I hope each of you feels empowered enough, important enough to pick one way to support research today, because your voice truly matters. And I do hope you come back in January and do it again. I want you to find an option that fits your family and know we are here to help. We want you to understand the opportunities, understand why they matter, and we want you to help sign up. If there's a barrier on that path, please reach out. Tell us that you need more information, you have questions, or you just want the researcher to email you first. Whatever it is, we are happy to help connect you to the opportunities available. And when we look to the future clinical trial pipeline, we need to be prepared as a community, scientifically and emotionally. We're going to give you the foundational knowledge of the process so you understand why we need to participate to validate, what a realistic timeline looks like, how many people have to participate, all the different ways to support that pipeline. KSF is here to provide all of that and in ways that are easy to understand and access time and time again. Importantly, I want to share that this pipeline isn't a one and done path. It's an ongoing cycle of learning more, finding those targeted treatments, and getting those treatments to trials and then to you. I don't have to tell you how different Kabuki syndrome is from person to person. So logically, each person might benefit from different treatments. The foundation is determined to find options for everyone, but we need everyone. Researchers and biotech companies ask us all the time, how many people have Kabuki syndrome? There are areas of the US and areas of the world where statistically we know there are people with Kabuki syndrome there, but we have to find them. When we say collaborating and knowledge sharing with researchers across the world, that also means finding the patients and physicians across the world that they're working with and the patients and the physicians that they should be working with. By being here, by sharing your experience, by supporting research, we can find them all and we can all work together. Lastly, research is finding treatments, drugs, supplements, new or repurposed, but research is also building a really strong foundation of knowledge so we know what to look for. So we know what symptoms to look for, and we know the best way to treat those symptoms, specifically for Kabuki syndrome. Better care can come from research, and that starts with a solid foundational education that we can provide with the help of our partners. I now want to welcome Dr. Hans Bjornsson to kick off the learning today with the epigenetic mechanism of Kabuki syndrome and the therapeutic development in mouse models.
Thank you so much, Amanda. It's my true pleasure to be here today and get to talk to the community. I'm a clinical geneticist. I work both at Johns Hopkins in the US and at the University of Iceland in Iceland. And I just want to thank the community. A lot of my research has been funded by donations and support by families. And in the 10 years that we've done Kabuki syndrome research, we've published about 17 peer-reviewed Kabuki syndrome patients papers, and I'm showing you 6 of these here. 3 therapeutic strategies. I'm going to tell you in more detail about one of them, and then 3 papers in green that have had mechanistic insights into Kabuki syndrome. And these are, I'm just highlighting a few of the 17. So I always start by saying, you know, we've had this book of life, the human genome, for more than 20 years. We finally have the full sequence, but we don't completely know how to interpret it. And the reason for that is that there are many different ways you can interpret that text because there are many different cell types. And some of these cell types are very different. You know, if you think about a sperm cell versus a neuron, they have very different components, and yet they have to read the same book, the instruction manual, the genome. And one way one could achieve this is by highlighting out the pieces that are needed by using kind of individual highlighting systems. But how is that actually done in cells? And I'd argue that one way to think about the epigenetic machinery is to think about it as the genome's highlighting system. It's a way to help pick out the areas that are most important to that cell at that time. So the epigenetic marks are these modifications of DNA or the associated proteins, and they're heritable through cell division. They're somewhat reversible, which is appealing to me because as a treatment target, and they're known to be affected by the environment to some extent. And they, if nothing else, they add to the information content of the DNA. And there's kind of 2 major groups. There's something called DNA methylation, a methyl methylation is added to DNA, or these histone tail modifications. And what the histone tail modifications are is that the DNA is wrapped around this thing called a nucleosome, the center here in the middle, the circle. And then there are these tails, protein tails, that stick out of that wrapping. And these tails can be modified in a way to decide whether the DNA is accessible or not accessible. And some of the marks are only seen in open chromatin, like these, H3K4 or histone acetylation. And H3K4 is really important for Kabuki syndrome, we think. So, you know, getting to treatments, you kind of have to have some understanding of the disease. And I started on this path. I thought the first clue came when the genes were discovered, and I'll explain why I think that's where the first clue came. So there are 2 different genes associated with Kabuki, KMT2D and KDM6A. The kids all have some developmental delay. They have immune problems and growth problems. But we didn't know the cause until relatively recently. So 2010, someone discovered that mutations in KMT2D are the cause of Kabuki syndrome type 1. And this is a highlighter that puts on this open chromatin mark H3K4 methylation. And then a few years later, someone discovered that an eraser called KDM6A that removes this closed chromatin mark, H3K27 trimethylation, also causes Kabuki, but Kabuki syndrome type 2. And to us, this seemed that there, you know, if you can't put an open chromatin mark, you may have difficulty keeping your chromatin open at the right locations. If you can't remove a closed chromatin, it's the same problem. You may stay closed. So that's kind of where we started for this first clue. And the Kabuki gene is big. This is data from Sid Banca's group. I don't know if we'll talk about this later, but you know, one of the questions is, well, does the enzyme function really matter? And what Sid did here, which was very clever, is he looked at all the mutations that were available and put them into these plots And he showed that individuals with Kabuki syndrome, they have a lot of these missense mutations towards the end, close to the enzyme domain. So I think this is the best evidence that the enzyme domain is important for Kabuki syndrome. So this is a really big gene, so it's too big for gene therapy. And it still has many domains that we need to work on to understand better. But the bottom line is, I think the enzyme domain is important. And to work on this, we characterized the first mouse model of Kabuki syndrome. We call it the beta geo mouse. It has a mutation that knocks out that enzyme domain in one copy. So it's similar to what we see in many of the patients. And the mice had a lot of the same things as our patients do. They were smaller than the other mice. And the mice even had immunological changes that were similar to what we see in patients. Here I'm showing you low IgA. That's something that we see very frequently in Kabuki syndrome. But the mice also had things that we didn't know patients had and haven't investigated fully yet. One finding that we found was that even though the bones of these mice are short, the growth plate is actually big, and that was a surprise to us. It must mean something that we're actively trying to study. Another thing that we found is that there's the immune system organizes in the gut to protect the body against all the bacteria in the gut. And there seemed to be fewer of these things called Peyer's patches, and they were smaller in the Kabuki mice compared to the litter mates. So that's another clue that we can use to go forward. So, we've been mostly interested in kind of the neurological phenotype, and we focused on that, and we've done a lot of different tests with these mice. But one of the tests that we keep coming back to is something called the Morris water maze. It's a tank, it's filled with water. We teach the mice to find an invisible platform. The mouse uses visual cues to find that platform. And then the last they remove the platform, and we see whether the mouse remembers where the platform used to be. And when we do that, what we find is the Kabuki mice in yellow, they cross about half as often as the other mice. So there's a memory that they're not as good at navigating this maze. And this led us to a potential clue. So when we looked into the hippocampus, which is the area that participates in visual spatial memory, and we looked at the brain, we saw that the Kabuki mice had fewer newly formed neurons labeled with green here compared to the other mice. So that's another clue that we could then keep track of. And some of this has panned out. So Jackie Harris and others have gone and tested this in patients and said, look, if we look at tasks where patients with Kabuki syndrome will participate in, and have to do with visual spatial, when we compare Kabuki in red to other kids with developmental delay, and then the normal population in blue, we see that the individuals with Kabuki have a lot of visual spatial defects. So that fits with what we found in our mice. And this is just another representation of the same data showing that this thing called block design, that's a task where our patients don't do very well. People in France, David Genevieve and Nathaniel Baudert, they have looked at the brains of individuals with Kabuki, and they find the same thing, that the area that we've been focused on, the hippocampus and the dentate gyres, they seem to be smaller in individuals that have Kabuki syndrome. So I told you in the beginning, we've done 3 therapeutic trials, but I'm just going to tell you about the last one. So this enzyme KMT2D, which is disrupted in Kabuki syndrome 1, it puts on this open chromatin mark, H3K4 methylation, but there's another factor called LSD1 that removes that mark. So our idea was that maybe you could inhibit this enzyme and get some therapeutic effect. And we collaborated with a big drug company called Takeda, and we were able to get an agent from them called TAC418. And when we looked in the brain of our mice that didn't get any drug, we saw, as before, a big difference between the mice that don't have a Kabuki mutation and the mice that do have the Kabuki mutation. We have fewer of these green cells. But on the drug, the Kabuki mice gained more of these newly formed neurons. And when we quantified this, we could see that there's a big difference between the littermates and the Kabuki without treatment, but a nice rescue of this measurement with this drug. Same is true for the behavioral testing. Here I'm showing you 3 different tasks. The first one is the same as I showed you before. You know, there's a big difference between Kabuki in yellow and the other mice in blue. But when you put the Kabuki mice on the drug, they do better at this task, suggesting that the drug is doing something in the brain. And this has led to a collaboration that you'll hear much more with Horizon and Jackie Harris working on an agent that they have that works in the same way called Wafidemstat. So, but I just wanted to say this is a therapy based on the first clue. It doesn't mean that there are not other ways that we can treat Kabuki syndrome, and I definitely think there's more research to be done. So just wanted to summarize, there are 2 types of Kabuki syndrome, and to me, it suggests that there's something wrong with the ability to open up chromatin. And our mice have phenotypic features that are similar to what we see in individuals with Kabuki syndrome. They have this hippocampal memory defect that we can measure in mice. And when we put the mice on this drug, LSD1 inhibitor, we get rescue of some of the phenotypes. So this is my group. I'm thankful for your time, and I'm thankful to all the people that work with me, and you'll hear from some of them later today. But I'm not going to take up any more time. I'm going to introduce Brittany Simpson, who comes from another stronghold of Kabuki, which is Cincinnati Children's. She's an assistant professor there and runs the Epigenetic Syndromes Clinic, and is a real dedicated advocate of Kabuki and active clinician caring for individuals with Kabuki. So thank you very much.
As Dr. Bjornsson said, my name is Brittany Simpson, and I'm a clinical geneticist and director of the Epigenetic Syndromes Clinic at Cincinnati. And today I am tasked with presenting, so today I would like to talk with you about the ongoing project that I'm involved with along with KSF that is addressing the need for a state of the science document. So this is a stepping stone to build bridges in Kabuki syndrome research. But before we talk about what the state of the science is, I wanted to outline how research is even funded here in the US. Each year, the U.S. government puts aside a set amount of money to go through the NIH, so through public funding, so that researchers can put in ideas for specific projects. And in 2021, that was $45 billion, which is a really large amount of money. But where does that money go? Well, out of all of the ideas submitted, which was about 59,000 proposals in 2021, only about 19.1% were actually approved for funding. And so further, this money, this large pot, is divided into different types of research. And when we look at rare conditions, about 13% or $6.2 billion is actually allotted to rare conditions. So how does that play out when we consider how many rare conditions there are? So about 30 million people in the United States, or about 9% of the population, is impacted by a rare condition. And we would anticipate that percentage would be similar around the globe. And there are about 7,000 individual conditions and counting. And if we take into account the amount of money that is set aside for rare disease, that's 6.2 billion, that means that about 885,000 is set aside for every condition each year, but that's only if it was equally distributed, which in reality it is not. But how does this compare with how much research costs? So the most common grant amount is $250,000 to $500,000 each year. And remember, only about 19% of ideas are approved when people submit for this amount of funding. This varies based on what the idea is and how the research proposes to complete the study. So costs are high and they vary widely. But then what about clinical trials, which I know is on the front of everyone's mind? Well, in rare disease, a phase 2 or 3 trial costs between $23 million and $174 million, with the median of about $64 million. So this is higher than in common conditions, which are the amounts you see below. And this phase 2, phase 3, that means we're testing the drug to see if it works. So we're past that safety phase. And so with this in mind, the limited funding, as well the competitiveness for funding and how much research actually costs, we as a community are poised to be able to act to improve the likelihood that researchers will study Kabuki syndrome. But not only that, we want to ensure they study a question that is answered, that has not been answered, and one that is important for individuals and families. So how do we increase efficiencies and guide this research to study Kabuki syndrome? We can continue increasing awareness of the condition and the needs of our loved ones and families. We can summarize what is known, and we can direct researchers to important questions to answer. And so to do that, we can provide scientists with a state of the science. And so what is the state of the science? It is a summary that determines the scope of what we know about Kabuki syndrome, what has already been investigated, and where are the gaps in knowledge. Maya Angelou said it well, that you can't really know where you're going until you know where you've been. Not only that, but this state of the science can really help researchers wade through the huge amount of information that is out there, which is a barrier. So just to give you an idea, in 2018, there were 2.6 million papers put out on all topics, so not just Kabuki syndrome. So that's a lot of information, and it keeps actually growing each year. So if we as a community are able to wade through this valuable information to summarize everything about Kabuki syndrome in one place, we can help increase the chance a scientist will want to answer the important questions that remain for our community. But you may be asking yourself, why does this matter? Again, it comes back to that point of limited resources and the fact that it takes a lot of time to summarize what is known, to put together an idea that you put in for funding approval. So put into the public funding mechanisms, put into other kind of private mechanisms as well. And remember, in the public world, only about 19% of proposals in general are funded, but that's the overarching percentage. Actually, other sections throughout the NIH are actually lower than that. And so with KSF, we are starting this process for the Kabuki syndrome community. And just to give you an idea what this has looked like so far, about 3,000 papers were found when we searched specific terms, which is a key point to remember. About 1,600 were actually duplicates, so we removed those because they were already in there. 123 were removed because they were just included incorrectly, which leaves us with 1,234 papers to review and summarize. So this is still a work in progress, so these numbers may change. But I mentioned earlier that it is important to know what you search matters. So these papers are placed in many different different kind of lists and journals and things like that. And if you search different terms, you may find different results. And so what the point is, you may miss things and not realize the study's already been done, or you may miss a past study that showed something that would really help your research along. Further, this amount of papers just go to show you the amount of time and effort required to summarize all this knowledge. I'm so excited that we have this really strong foundation, but now it will be important to condense it down into a manageable amount of information for scientists to use to answer the questions we need answered. And not only that, but there is another important reason for our community to do so. We want to make sure that we are guiding them in the right direction. A lot of times scientists want to study what is clinically relevant, most of the time, in fact, but they may not have the clinical knowledge from direct patient care to truly guide them. They mostly find this information by reading a paper, and so they may not fully understand what is an important area for patients and families. So this is an opportunity for us as a community to identify what those research areas are for those scientists. And so, as a community, we are very powerful and have many roles when it comes to research. We need to continue to increase awareness of Kabuki syndrome, as well as continue advocating for those research gaps. And importantly, the community can facilitate research through the scoping review that we have talked about to save researchers' time. Also, and vitally, individuals and families can help research by participating and engaging in studies. We cannot do this research without you, and we greatly value the time you've donated thus far for these endeavors. Further, and unfortunately, the reality is this stuff costs money, and the public money is limited. So fundraising, as KSF and other groups do, is very important. And again, through all this, we can help guide these precious resources and prevent duplication of efforts to drive research to the areas where the gaps are. And KSF has already started to identify these areas of importance with last year's survey. So last year, if you remember, you as a community spoke, and we will be comparing these voiced needs with what is already done to see, what differences there are. And so look out for more to come in the coming year regarding this. And so last year we went over this, and it is still just as important. So we want to be sure all our research has the patient at its center, with collaborations to drive research as physicians and researchers, drive research where it needs to go. With a patient-driven approach, we attempt to collaborate as physicians and researchers to allow for a better understanding of a patient's journey over the lifespan, to allow for meaningful questions to be answered. When we find those questions, we can research those, which takes a lot of time. I'm sure you guys are aware, but we hope to find valuable information for patient outcomes and qualities of life. But involving the state of the science in this pathway further directs those resources to the areas of most need. We then can facilitate our collaborations with our wonderful researchers and physicians, and can hopefully help to decrease the time to findings to address and improve quality of life. And with that, I would like to thank you for your attention, and please let us know if you have any questions. Our contact information is here. If you would like to visit our team, we'd be more than happy to see you and your family. And with that, here is my rockstar team, which includes Dr. Diana Brightman, who is a genetic counselor with a PhD, whose science involved epigenetic regulation. So she is a key asset. And our lovely clinical research coordinator, Deemer Quad, which some of you may have met or talked with. She is also a budding physician. And so thank you again. And without further ado, I would like to introduce Corinne Stobaugh, KSF Research Advisor, and importantly, mother to Gabe.
All right. Thank you so much, Brittany. Dr. Simpson, let me go to presentation mode. All right. So today I'm just going to be talking to you guys about the project I'm doing with the Kabuki Syndrome Foundation. Last year, KSF asked if I could help with a project that involved categorizing the medical literature on Kabuki syndrome, and the preliminary results were utilized alongside data from the crowdsourcing survey that a lot of you participated in to compare topics covered in the medical literature with topics deemed important by the Kabuki syndrome community. We had a lot of results from there that were very exciting, and I thought if we could generate useful information from a kind of informal Excel database, what could we accomplish with a more formal one? I work at the James A. Haley VA Medical Center in Tampa, Florida, and our medical library has access to a citation management database called PicoPortal. And it has some unique features that I thought would be helpful in this endeavor. It's a web-based application, so users don't have to download licensed software, and the data can be accessed from anywhere. Members can be added to a project from different institutions at no additional cost. You can store PDFs for citations, so full text is always accessible to any user, regardless of institutional licenses. You can add tags and keywords to categorize and organize all the articles and citations, and you can create tailored data extraction to identify key elements of a citation specific to a research question. So why is this important? A lot of research questions start with a literature search, but the problem is that there are multiple databases that contain published research, and results are often dependent on how you perform the search and how articles are categorized within the database. For example, abstract posters presented at a medical conference can take a lot of time and effort to produce, but sometimes these efforts don't result in a peer-reviewed publication the data is kind of buried in a large abstract symposium. We want to make sure all research efforts are cataloged in an academically rigorous manner, and make sure that anything that could be useful to the community is captured. Also, PicoPortal saves all the search parameters. So if you're a Kabuki syndrome researcher and you have a research question, your first real step, a literature search, is kind of already completed for you. The current projects being facilitated by the database include State of the Science, which Dr. Simpson just talked about, and development of Kabuki syndrome clinical guidelines. In the future, we hope that the database can continue to serve the ultimate goals of forging collaborations among researchers and the community to utilize the limited resources we have in the most effective way. We would now like to open up the floor for a question and answer session, which will be moderated by Amanda.
Thank you, Corinne. Brittany, Hans, if you want to rejoin us, we can get started on these questions. Thank you guys for submitting your questions. At any time, you can submit a question in the Q&A right next to the chat. You can also head over there and upvote the ones that you're most interested in hearing today, and we'll do our best to get to all of them. The first one is, what is a growth plate? Hans, I believe that you said the bones in mice were shorter, but the growth plate is large. What does that mean? And are there potential opportunities for humans with Kabuki syndrome based on that?
Well, the growth plate is basically the site where the bone is expanding. So, it's like the zone where cells are very active, and then the growth plates close when people stop growing. And, it was a big surprise to us to see that the growth plate looks expanded. It may mean that some cells are dividing more and some less, and that yields the smaller bones. So we're actively trying to work on this, but I think it's a clue that could help us get deeper into the kind of mechanistic basis.
Thank you. Switching gears, several people were curious what type of treatment or therapy is done for visual spatial difficulties.
So, sorry, who was this question for?
Sure. I think in general, whoever wants to field it, or I can field it if you want. I think they're asking about current therapies for visual spatial, which is actually something that we will cover later this afternoon when we look at neuropsychological testing and other school supports. If you want to field that one, now we can, but we also have some other popular ones as well.
I think people will know more about this after hearing Dr. Harris speak, but I think, kind of mapping out, strengths and weaknesses is super helpful for educational activities as well. And, so it's important to map that out for clinical trials, but it could also be quite useful as we provide guidelines to teachers or educators or others that work with individuals with Kabuki syndrome. So I don't know that we have a specific therapy, but you'll hear more about that by Dr. Harris.
The next question that we have, when we look at intervention approaches, will it be different depending on if it's type 1 or type 2 of Kabuki syndrome? And when people ask that, they're referring to type 1 being a mutation in the KMT2D gene and type 2 being the KDM6A gene. So will we need different therapeutic strategies depending on the type?
Go ahead, Hans.
No, go ahead. I don't want to hog up all the time. You
Oh,
should-
no, you're fine. You're fine. But I mean, it just, Dr. Bjornsson may have a lot more to say as well, but I think that there will be likely similar strategies for both, because these kind of machines in the body work together to perform similar activities, but there also may be different ones just because they are a little different. So I would say both.
Great, thank you. Let's see, our next most popular question is, given that other LSD1 inhibitors are on the market right now, do we know some of the typical downsides of those drugs, like side effects? And again, you guys are asking great questions. I would argue that our audience is ahead of the game and looking forward to other presentations as well. But Brittany or Hans, do you guys want to field this one?
So I think the reason we're excited about Horizon is that they've already used that drug for a lot of people, and there's been relatively limited side effects. So I think we feel pretty confident that it's a safe drug. We hope it's going to work in the clinical trial and do something, but it doesn't seem to be massive side effects, is my understanding.
Thank you. There are a lot of questions. I'm trying to be fair here. If we look at LSD1 inhibitors effects being similar to HDAC inhibitors, so these are two different drugs basically, could you compare the two? I feel like that's a very scientific question, but it definitely leads to some educational opportunities for Hans and Brittany and the foundation as well.
The HDACs in general are pretty toxic agents. They're very broad acting, and the trials we did, I look at them more as a proof of principle that there's something that could be done. I think it's hard to move an HDAC inhibitor. There's this natural HDAC inhibitor, which is the ketone beta hydroxybutyrate, and I think that's probably our best bet. But a lot of these agents are mainly used in cancer. I think the histone methylation agents seem to be better tolerated and more specific. So I think that's why we've kind of moved towards that route.
Thank you. And for Corinne, the research articles that you shared, are those articles available to the public, and do they have layman summaries?
They are. Some of them are free full text that you can, download. There's the most common database is PubMed, and that's available to anyone. And you can do medical literature searches, and depending on how they were published, they may be open source. But anybody who's interested in one that there's like a paywall, you can email me and I can give it to you. I pretty much have all the PDFs for every Kabuki syndrome paper that's out there.
Awesome. Thank you, Corinne. And that is something, that's part of making research accessible as a foundation. If there is a really key publication, we're going to really put that on our website. You can find some of them now. And I think adding layman summaries is a great suggestion. We're listening to suggestions like those, so please do share. I have time for maybe one or two more questions. There was a really popular question about genetic testing during pregnancy for Kabuki syndrome, and that's really, really going to vary by your clinic, by your state. It's something that we can talk more about offline, depending on where you are. Feel free to shoot me a message. I want to get one more question out here for the researchers, and it is, when we look at epigenetics and the new studies that are coming, do we really think that we can reverse some symptoms? And also, are we considering the epigenetic impacts that come from the environment in terms of PFAs or other hormone disrupting chemicals?
So I think that's a very insightful question. So there are going to be things that have been developed a little differently because either that KMT2D or that KDM6A machine wasn't as efficient as a child was developing in mom's stomach. So there are going to be some things that may not be changeable. But after a person is born, those machines are very important, as we all know. So some of the things we can impact with medicines. Sorry, I've forgotten the second part of the question. That's a very, very good question. Some things we won't be able to fix, but some things we will. So we need to really stress the things that we can impact.
Great. Thank you, Brittany. We are short on time now, so I am going to pass this to Corinne. Hans, Dr. Bjornsson, Dr. Simpson, thank you so much for joining us this morning. Be sure to check the agenda so you can interact with them later as well. I'm going to pop off. I'm going to leave Corinne on, and Corinne is going to introduce our next segment. Thank you.